Good afternoon, everyone. Thank you so much for joining us. We are going to give everyone about 1 minute just to get a few more participants logged on. So bear with us for just a moment and we'll get started shortly. Just a few more minutes and we'll get started here. All right, well, welcome again. Thank you so much for being with us today. My name is Hannah Huber. I'm a global product manager for a healthcare group at Adhesives Research. Really excited to share some content with you today that we feel is timely and relevant to some of the challenges you may be working through. So we'll get started here with just a few brief comments, but you're here today for a webinar on how pressure-sensitive adhesives can improve rapid scale-up of your microfluidic devices. We have a couple things to run through and we'll make it brief, but just a little bit of webinar housekeeping. I'll provide a brief introduction on why we wanted to share this content with you today, followed by our presentation by Tom Gordon. And I think you'll be really pleased with the content there. At the end, we'll have a Q&A session and I'll walk you through how that works as well. So just some housekeeping for you. The participants will be in listen and view mode only, so you will not be able to activate your webcam or your microphone. The question and answer session will be held at the end of the presentation. Please submit your questions through the Q&A feature. We understand there's also sort of a chat function that you may be able to access, but we ask that you please use the Q&A button. That will help log all the questions in an orderly fashion. If we don't have time to get through every question today, we will certainly follow up with you directly. You are allowed to send anonymous questions and you're allowed to view all questions, but you cannot upvote. So again, any unanswered questions will certainly be followed up and the webinar recording can be accessed on demand after this event. And then please use the raise hand feature if you need any special assistance. So we wanted to just frame for you why we felt this was relevant today. We have seen a very large growing demand for microfluidic technology and devices in point of care diagnostics, with a growth rate of 11.25% projected from 2020 to 2027. We are all kind of living and breathing in a different time right now. And I know that you are feeling the pressures of those demands on your business to deliver these technologies. So some of those drivers changing sort of the climate, if you will, is the increasing incidence of disease. I think we are all very much aware of that. The increasing demand for low volume sample analysis, desire for multiple analyte detection in a single device, and then of course, faster turnaround time. Some of the constraints that you may be, or restraints you may be experiencing are integration and certainly cost. And I think one of the things that we wanted to capture today are some of the challenges that we see that are common among our customers. There's certainly been a shifting focus of project priorities during this global pandemic. Adapting existing platforms for COVID testing is another theme we understand that our customers are struggling with and wanting to gain some speed to use existing technologies. Of course, developing new COVID platforms as well. And then looking for solutions to meet new objectives very quickly. So speed is a theme that will recur throughout our talk today. But we also understand that you're managing some existing pain points and looking for materials that can do a number of things. They need to be biocompatible. They need to bond to dissimilar surfaces. You're looking for materials that have low residuals. And obviously we are looking for some options to provide low autofluorescence if that is one of the key measures in your device. Delivering thickness control can be extremely important if, especially with spacer tapes. We understand the challenges there and the need for consistency from batch to batch. And we also know that you're looking for precise contact angles to manage the fluid flow through your device and for materials that enable production. We get a lot of feedback about some of the challenges that we may, maybe you've found the perfect combination of materials, but you get into scale up and you discover some challenges there. So we definitely are aware of that and are working and designing products that will help aid that process instead of inhibit it. Just a few final comments before we get started. I think one of the best takeaways that you may get from today will be the question and answer session. So I hope that you can spend some of the time during Tom's talk to think about what you may want to ask. Best case scenario today is you walk away with an answer in real time that you can take to your teams and to your projects that will help you right away with some of the challenges you may be experiencing. In addition to what we've outlined as what we see as common issues or pain points, what is most interesting to us is likely what is most challenging to you, which is likely what is most unique to you. So there may be some things that you want to raise to our attention that are outside of sort of these kind of common pain points listed here. And to be frank, that's really the joy and the motivation in our work, is to solve those unmet problems and deliver the solutions to you. So we hope that you really do take to heart our interest in engaging with you. Again, I think you'll find the question and answer session to be as valuable as the content we're about to deliver. And again, if we don't get a chance to answer your question today, or you don't have one for us today, please do reach out. We would love to work with you and help you take this project that you're working on to the next level. So thank you again for your time. We really do appreciate you being here. It's a chance for us to learn too. So that is very valuable to us. And without further ado, we will get ready.
Hello, my name is Tom Gordon. I'm the Healthcare Innovation Group Leader at Adhesive Research. Today I'm going to be discussing the rapid prototyping of microfluidic devices with pressure-sensitive adhesive tapes. So Adhesive Research has a 25 plus year history of providing tapes to the medical device industry, including in the area of microfluidics. We began with developing tapes for lateral flow immunoassays, so things for pregnancy testing, for infectious disease, as well as for the identification of chemical contaminants. We then moved into the area of microfluidics and devices such as blood glucose test strips, blood coagulation devices, the detection of prostate-specific antigen, as well as forensics and drugs abuse. And we continue to innovate for the future in areas such as continuous glucose monitoring, gene sequencing, PCR testing, and organ-on-a-chip or cell growth types of platforms. And before I get into the rest of the presentation, I'd like to briefly review the types of pressure-sensitive adhesive tapes that are available in the marketplace. So one would be transfer films, in which a single layer of adhesive is placed on a release liner. This can be used to bond two materials together, and this is your best option for forming a very thin channel height with a pressure-sensitive adhesive tape. There are also single-face tapes in which a polyester film or another type of backing can be used to support the tape. These would be bonded or only on one side, and they would have a release liner protecting the adhesive. And then there are double-face tapes in which the carrier has adhesive on either side, which is protected with a release liner. Microfluidic devices are commonly prepared through a process known as soft lithography. It begins with the traditional photolithography approach, in which you take a silicon wafer, you spin coat SU8 or another type of epoxy material onto it, and then through a lithographic mask and UV light, you form your master device. So this type of master device can then be replicated numerous times in the soft lithography process. And that involves essentially casting PDMS onto the surface of the master mold, curing it, and then removing it from the surface. and then applying it to a glass slide surface after plasma treatment. And this is a highly precise fabrication method. However, it requires this microfabrication facility, which is expensive to access or expensive to build, and requires that plasma treatment in order to get good bonding. And altogether, it's costly and time-consuming process. So researchers have developed tape-based approaches to do rapid prototyping of these types of microfluidic devices. And that can begin with the design of an AutoCAD design of your microfluidic channel. That can then be easily cut with a plotter cutter, which is often referred to as xerography, using a razor to cut the shapes of the microfluidic channel, or through laser cutting of a tape into the appropriate microfluidic channels. That tape, that after it's cut to the appropriate microfluidic channels, can then be assembled with a base and a cover to form your microfluidic device, simply with the application of pressure to bond the adhesive. And then finally, once this full device is formed, it can be disinfected or primed in whatever way is necessary prior to the introduction of the biological materials. A recent report from Rothbauer and et al. In Scientific Reports discussed the characterization of 4 biocompatible pressure-sensitive adhesives for this tape-based approach for lab-on-a-chip and organ-on-a-chip systems. And if you look at the approach that they're taking, it uses much lower cost equipment relative to the microfabrication path that I discussed on the previous slide. It can cost $1,000 to $20,000 for one of these home hobby types of plotters, and up to $100,000 for a more professional, more capable instrument. And you can also get into laser cutting with between $20,000 and $150,000 worth of investment. So it's not too hard to get the equipment and not too expensive to get the equipment required to cut these microfluidic channels. And I think even a better advantage is the total time spent between designing the AutoCAD file to producing your microfluidic device and disinfecting it is a total of 1 to 3 hours, which is substantially lower than what is required to access the microfabrication facilities and produce the full device through soft lithography. Other interesting findings are that acrylic-based PSAs were found to have lower vapor and moisture permeability than PDMS, which for certain applications such as organ cell growth is very important. You don't want to have very high moisture permeability. Acrylic adhesives were also found to support cell growth on their own, which is a problem for other types of materials used in microfluidic devices. And then the membranes and scaffolds can be introduced into the device on their own through the use of the PSA tapes, which allows for the support of organ cells and other types of cell growth. I think another major advantage of this type of approach is it allows for the bonding of dissimilar materials. So PET, PDMS, PMMA, COP or COC, all these materials, polycarbonate, polystyrene, can all be bonded using pressure-sensitive adhesive tapes, as well as the membranes and scaffolds, as I mentioned. And that can provide a huge advantage. For instance, I show an example here where polycarbonate can be bonded to polydimethylsiloxane or PDMS through the use of a differential adhesive tape, where an acrylic adhesive is designed for the bonding to polycarbonate, a silicone adhesive is used for PDMS. This is something you can't accomplish using that traditional approach. However, there's some very critical aspects of a PSA tape that need to be taken into consideration when you're selecting a tape. The first would be the biocompatibility of the tape. Adhesives can be tested for their accordance with ISO 10993-5 and -10, cytotoxicity, skin irritation, skin sensitization, and that gives you an idea of whether the tape will support or interfere with biological activity. Related to that is the residual content in the adhesive. So after the adhesive is manufactured, it should have low residual content of volatile materials. And I show an example here of R-Care products, acrylic tapes, relative to a competitive tape, showing the low outgassing that we can achieve in our tapes by designing the process to produce them in a way that's going to drive down residuals. And in addition, the uniformity of the tape is very important, particularly for microfluidic devices. Your channel height is essentially defined by the thickness of your tape. So specialized coating equipment is needed to get very uniform thickness of adhesives for this type of application, and repeatable adhesive thickness as well. Another important aspect to consider is moisture resistance. Adhesives should provide a moisture or fluid resistant barrier. Particularly, this is important in multi-channel devices where you do not want fluids to be interchanging between the multiple channels of the device. That also includes, you don't want air or other contaminants from getting inside as well. Autofluorescence is also very important to keep in mind if you're doing an optical type of test. The tape cannot fluoresce excessively in the region of interest. So we show here numerous adhesives produced by Adhesives Research showing that they have relatively low autofluorescence at some common wavelengths of excitation, 485 nanometers and 532 nanometers. And then finally, the cuttability of the tape is very important. So if you're going through either a plotter cutter or other types of cutting, laser cutting approaches to define your channel, the modules of the tape is very important to consider that it's going to cut well, it's not going to smear on the edges, and it's going to make a very well-defined edge for your microfluidic device. And a real advantage of this rapid prototyping using tape approach is that it's also easily scaled. So you can take that same AutoCAD design that was developed for use in laser cutting or the plotter cutter and introduce it right into medical grade converting, in which you would cut the tape roll to roll and wind up a converted roll that has your microfluidic channel already cut into it. That can then be used in a reel-to-reel type of manufacturing environment or reel-to-chip manufacturing environment in which all the required components for the microfluidic device can be added in a reel-to-reel environment. And that is the best approach to do large-scale production of microfluidic devices. And the various options to cut these channels would be things like laser cutting, which I mentioned earlier, as well as rotary die cutting or flatbed die cutting, which can cut the same shapes as produced with these other approaches. Adhesive Research provides some really unique technologies in the area of microfluidics. One would be our hydrophilic pressure sensitive adhesives. So these are really unique products to provide both pressure sensitive adhesive strength as well as hydrophilicity, which allows for fluid flow and wicking of fluids into the channel. So we provide both a 93049, which is a single phase tape, and a transfer film 93450 for these types of applications. We also provide high uniformity double phase spacer tapes to define the channel of a microfluidic device. So these include various different acrylic adhesives, heat sealable adhesives, as well as different thicknesses of carrier that can be used to define your channel height as needed for a particular device. In addition, we offer numerous transfer films at various thicknesses. So these can be silicones, these can be acrylic-based tapes that provide very strong bonding to dissimilar materials, as I mentioned earlier. We also offer products that are optically clear, so they can provide a very clear path for either identifying fluorescent compounds inside the material or looking at fluorophores, or for exciting those materials as well. In conclusion, adhesive tapes provide a low-cost platform for the rapid prototyping of microfluidic devices for a multitude of applications. However, medical grade tapes must be properly selected, and there's some important characteristics to consider, such as the biocompatibility, the residual content, the thickness and uniformity, the moisture resistance, the autofluorescence of the tape, as well as the cuttability of the material. And these tape-based microfluidics can be easily scaled to high volume, reel-to-reel processing. Adhesives research provides a lot of value to the customers who partner with us. We provide design flexibility, we provide very fast development capabilities, and it leads to extraordinary outcomes for our customers. And so, our R&D department partners directly with our customers, the engineers who are designing microfluidic devices, so we can provide the ultimate solution that will solve your problem. And at the bottom of this slide is shown some of the manufacturing and production sites that we have, both in Pennsylvania, in Ireland, and we have sales offices in China, and we also recently brought online medical converting in Glen Rock, Pennsylvania as well. Thank you for your time, for being involved in this presentation. I invite you to contact us at Adhesives Research. We'd love to partner with you to solve your problems in microfluidics, and we have a lot of unique products to offer in this area. So thank you.
Great. So we can now begin the question and answer session. If you have any questions about the talk or about just general microfluidic needs, there's a Q&A feature at the bottom of your screen. Click on that and you can type your question in. So I know one question came in prior to the presentation, so I think I can address that one first. It was a question about considerations for adhesives that are used in thermocycling, specifically PCR, and then how thin can a double-faced tape be made, I assume, for a similar application. So I think the important, some of what I talked about in the webinar would be some of the first things. So all the biocompatibility, the low residuals, all of that, those are, and autofluorescence is particularly important in PCR. So all those aspects you still need to consider when selecting a tape for PCR testing. But the real key with PCR is you are having a lot of temperature swings. So going up to 90 to 100 C, and then dropping back down to a lower level. So the best adhesives for that are resistant at that high temperature. So that can be acrylic tapes, silicone adhesives, or acrylic adhesives. The one advantage of silicones is that they have a lot more stable modulus relative to other acrylics and other types of adhesive systems. So while you're fluctuating between 60 and 90 C or even room temperature, you have a lot less of a change in that silicone material. It tends to have better bonding over a range of temperatures. So I would say silicone is a good option. But we have acrylic options that can work as well. And then in terms of the second part of the question about how thin you can go, we're always trying to push the limits of what we're capable of doing. I would say something that would be comfortable for us is something like a 12 to around 12 microns, 12.5 microns thick of PET, and then 12.5 microns of adhesive on each side. So making a total of around 37.5 microns. I think we could push that lower if that were needed, but that's sort of a comfortable level for a double-faced tape. So there's some questions here about thickness limitations. So I sort of just addressed that in terms of we can go down to about, I would say 12, 10. So adhesive alone as a transfer film, we've gone lower, 10. We've had some customers interested going down to 5 microns in adhesive thickness. One thing to know about when you go to lower thickness is you also tend to reduce your adhesion or your bond strength. So that's something we need to consider as well as we go to lower thickness. On the high end, we can coat it on a lot of different carriers of different thicknesses. So, depending on your need, we can do a thicker carrier, up to 100 microns, 200 microns, or even higher, and then various different adhesive thicknesses as well. Whatever is needed for the application. So what is the difference between standard products available in market and custom-made products from Adhesives Research? Yeah, it's a really interesting question. So we have the capability of really customizing all aspects of the adhesive. So whether it's the thickness, whether it's the type of carrier, whether the carrier is white or clear or black, we've done all those things. We can screen all the properties, so autofluorescence, all those types of things. We can also, so it's really completely customizable if you have if you know what you need and you're testing something that's working okay, but it's not working in a certain aspect. We have the capability of matching properties, but then improving the one aspect that you need improved. And then I would also say that some of the things I talked about today in terms of residuals, in terms of uniform thickness tolerance and repeatability, I feel that we outperform our competitors out there, where we're really trying to make the cleanest tape possible, very low in residual content that's going to have a chance of interfering. So I've heard from customers who have had challenges with other acrylic tapes, and then they use our acrylic tapes and find that they don't have any issues. And it could be because there's a lot of residual monomer in those other tapes. I guess those are a few things. I don't know, Hannah, if you had anything else to add on that question.
Just that I think the ability for us to engage with you to solve that specific problem, like that's really the main difference, is that if you found us, you probably have already worked through some standard products that are otherwise available to you. We really do tend to fill a gap for a lot of customers who have exhausted other products and are really stuck with a handful of challenges that they still need to solve, and so we absolutely welcome those challenges, and are really willing to focus on what is unique about your product and what we can deliver to get you across the finish line.
Next question here is about oozing for this product. I'm not sure if there's a particular product you had in mind, but yeah, so oozing or creep of the adhesive within a microfluidic channel is certainly a concern. You need to look at things about, what temperature is it exposed to, and then what is the modulus of the adhesive, and does it have a tendency to creep at that temperature? So, we do have in adhesives there additives like cross-linkers that help to tie the polymeric network together. It's going to prevent some of that creeping from occurring. I'm not sure if there's a particular product in mind there, but yeah, we certainly have characterized it, and if we know the temperatures that you're using, we can have a good idea of whether you're likely to see creep or not. Expand on the manufacturing process using roll to roll. So yeah, so in our process, it's really so we do everything roll to roll. So we start with whatever substrate or maybe a release liner that we're coating our adhesive onto. We're coating the adhesive, let's say, onto the liner, drying off the solvent that may be used to disperse the adhesive, and then laying in the substrate, winding it up, making a roll. It would then head into converting, where you're cutting out the microfluidic channels. Excuse me. And then at that point, it can be rolled up into, you're making discrete parts that then can be rolled up. Beyond that, at our customer sites, I mean, there's a lot of uniqueness, I would say, in how our customers then make their final microfluidic devices. So there can be liquid dispensing that goes on throughout that process. There can be different printing steps where certain conductive components or different biological materials are printed onto the surface. So there's a wide range. It sort of depends on exactly what type of testing you're doing. I mean, there's some applications where it's relatively straightforward and simple, and then there can be very complex devices made roll to roll. So I'm not sure if the question was more in terms of the microfluidic devices themselves or in our process, but hopefully that gives you a little bit of flavor. So how tailorable is the adhesive's hydrophobicity? Yeah, that's a great question. So like we mentioned, we make hydrophilic adhesives. In terms of contact angle, something that's really hydrofluidic will have a contact angle that's less than 10. So that's basically wicking behavior. It's going to suck the fluid into the channel. However, we've had some customers who really want a tailored contact angle or level of hydrophilicity. And I would say we do have the capability to adjust the hydrophilicity as needed. So, a typical acrylic adhesive will have a contact angle more like over 100, 110, something like that. But we've made tapes that are at 20 or 60 or 80 degrees if there's a specific need to require that particular contact angle. So yes, it is tailorable.
Tom, is it the same for hydrophobic materials?
Yeah. I mean, hydrophobic would be more like that 110 degrees of contact angle. And certainly we can go, higher as necessary for a particular application. Any tricks to help wet out of the adhesive to remove air bubbles? Yeah, so I would recommend anybody who's bonding pressure sensitive adhesives to get a roller, like a handheld roller if you're doing it by hand. You lay one end of the adhesive down, put your roller down, and then roll the rest of the adhesive onto your substrate. That's going to be your best way of removing air during application. You can also look at getting like a laminator in, simple two rubber rolls that are connected together. You can control the pressure being pushed between the rubber rolls and set the speed. That gives you a little bit more control over it. Instead of just applying hand pressure. But that's the best way to eliminate bubbles, I would say, during lamination, is getting a roller. That'll really help you out. So here's a question about how and where can I buy your product? Hannah, do you want to take that one?
Yeah, sure. We do sell direct to our customers, so please contact us through our website, or you can reach us at marketing@arglobal.com. Let us know what you're looking for. You may not know what specifically, just if you could give us some detail on what challenge you're trying to solve, what you're trying to bond. We will follow up with some additional questions likely to help identify what the right product would be. We would then sample to you what we think would work, and kind of work through some feedback with you to make sure we've landed on the right selection and supply you directly from either our Glen Rock facility or Limerick, Ireland.
Great. Yep. There's a question here about how do you evaluate or measure the adhesive uniformity over a surface? How much air is trapped between the adhesive and the surface? So I guess the first thing I would say is, we strive to have a very smooth adhesive surface, but obviously there could be some fluctuation on the surface. I think this kind of points back to the question of how you're applying the adhesive. So if you're getting lots of bubbles, may not be applying it with a roller. If you are, you may want to look at how you're applying the pressure and that type of thing in order to eliminate bubbles if possible. But we also have capabilities of understanding like how much surface variability there is. So, how much, if there's any peaks and troughs, that type of thing on the surface of the adhesive. So there are ways to do that, and I'm happy to talk about how much variation we see in that as well. So if there's any more specific question on that, please follow up. So I think this is a question asked, or I think I had earlier about thermocycling and PCR applications. And then, can you supply sheets instead of rolls? Yes, we can. So we often start with sending sheet samples to our customers. We also have smaller rolls that are available to purchase, 15-foot, that type of smaller size rolls, 9-inch wide, that type of thing. And we have a lot that are available for quick sampling.
Tom, we can also provide sheets at commercial volumes as well.
Oh, right, sure. Yep. Yeah, that's a good point. So yeah, we can do the whole converting process to make microfluidic channels and then sheet those pieces. So you're instead of being sent a roll, you're being sent in individual pieces. Yep. Thanks for adding that. Is there a typical shelf life for your PSAs? So yeah, typical is 1 year, and then Yeah, so this specifically asked about 93049. So typically it's 1 year. That's what we, I guess, would say certify the material for. We've certainly had people use the material beyond that shelf life and have success. 93049, yeah. I actually think that so there's some products that have longer shelf lives. I can't remember if that one is off the top of my head, but we can follow up on that. And then the specific questions about minimum order quantity. Yeah, so yeah, I think typical is a year. I don't think we have many products that go beyond 2 years. So typically 1 year.
We can provide assistance as needed if you have a unique situation and just need to know that your material is still in spec. We are able to support you, in certain instances. It doesn't mean that we can extend shelf life or warranty material beyond shelf life, but we can certainly help you work through whatever specific challenge you have based on expiry. It's typically just making sure the product's still in spec. So.
So there's a question here about developing membrane flow through a device and would like to bond nitrocellulose to polystyrene frame with an orifice for sample application. What tape would be a good option for bonding these two surfaces and for converting capabilities? Yeah, so I think we can make some suggestions there. I think something like 90106 MB would be a good option. But we have some other ones I think we could sample to you as well. I don't know if you have a specific thickness that you're interested in maintaining for that, or whether it's just a general bonding question. But yeah, and certainly it would convert well. We have lots of people converting that tape, so it should be fine. Are there adhesives which will bond to metallized plastic? So yes, sometimes we use metallized plastic as a carrier. So like MPET or other metallized polypropylene, that type of thing. Worked with that before, so yes, we definitely have adhesives that bond to metallized plastic. Say you have tapes designed to passively control reduced bubble formation via surface coatings and our semi-permeable membranes. Ooh, that's a tough one. I'm not sure we have one exactly designed for that. I think I need a little bit more information about exactly what you're doing there to answer that question properly. So I mentioned our hydrophilic adhesives. They certainly allow for good wetting on a surface. So I'm not sure if that could help with reducing bubbles that could form. But yeah, maybe provide more info on that one, and we can help better, I think, address your question. And typical contact angle for a silicone adhesive. So over 100, I can provide you a more specific answer, but I don't have the exact answer on the top of my head, but it's certainly hydrophilic or hydrophobic. Okay, so I think actually got through. Oh, and there's a question here about typical thickness tolerances that I missed. So typical thickness tolerances are mostly around plus or minus 10%, but we can typically do a lot better than that. And we've had for some products as tight as plus or minus 2.5 microns. Some of it comes down to our coating techniques. We have some very specialized coating techniques. If the need is there and the volume is there, we can really tighten up the thickness tolerance. But it does sort of depend. For our current products, it really depends on the product, what was needed when it was developed. But we have the capability to really tighten up the thickness tolerance when needed. All right. So,
Tom, I'll just add if we're given a few more minutes, I think we have maybe one or two more questions time for if anyone wants to submit a last question. But while we're waiting, I just want to thank you again for joining us and let you know that, again, one of the things that we maybe do differently than some other groups out there is we really do provide access like this to Tom and his team. And he's not alone in being one of many talented members of our team that does work very closely with you. We don't put up a lot of gates. We really try to make that conversation as accessible as possible, and make sure that you're getting the technical support that you need. So again, if you do have other questions or concerns that you could share with us after this session, we hope you reach out to us. We look forward to hearing from you. We really do enjoy the challenges that you bring to us. So please don't hesitate to reach out, and we promise we won't keep Tom from you. We'll get him engaged right away.
Great. Yeah, I didn't see anything else come in, so I think we addressed the questions that are out there today. So I want to thank everybody for joining this question and answer session and listening to the webinar. Like Hannah said, please contact us and be happy to engage in your application.
Hey, Tom, real quick, I see one last question that snuck in there. We've had great success at laser cutting your PSAs. Do you have any recommendations for minimizing any edge burrs generated as part of the cutting process? Great question.
Yeah, did you say laser cutting?
Yes.
Okay. Yeah. Oh, I see it here. Yeah.
Laser cutting. Yes. Mm-hmm.
Yeah. So for laser cutting, yeah, so it does depend on the material. We've seen that, like, a polypropylene versus a polyester or polycarbonate, they all cut a little bit differently. So I know there are a lot of different settings with the laser cutter that you can adjust. So things like the power, or even if you can try a different wavelength that could behave differently. So yeah, I think we can provide some more suggestions. We have some more people internally who I think have more experience in that area that I think could provide a better answer than that. But yeah, what I've seen is that customers will come to us, say they're getting burning or burrs or that type of thing, and through adjustment of some of the settings in laser cutting, they're able to overcome that issue. But one of the things that can be more of a challenge is if you're dealing with lots of different materials, each of which cuts differently, that can be a bit of a challenge. All right.
Thanks, Tom.
Great. All right. Well, thanks everyone again for your attention, and please contact if you have any follow-up questions. And we'll be following up with everybody, I think, after in the next coming days as well.